Okay, so I'm Andy Lee, one of the co-founders of Vincere Bio. For those that you don't know, today we're going to be presenting this new paper which came out last week from Steve Horvath's group looking at universal DNA methylation age across mammalian tissues. This group produced one of the first DNA methylation clocks showing that you can look at differential methylation sites and predict a biological age. And this is now a continuation of that work that is continuing to evolve. While the prediction of biological age is itself interesting, we've been able to extend on some of these methods, starting to look at disease state as well. And I think that's super interesting that DNA methylation changes not only with age, but also with disease. And so I think this is one of the new components and tools in the toolbox of multi-omic biomarkers that can really help to hopefully change the way we do medicine with earlier diagnosis and with better tracking of actions. So key points from this paper: DNA methylation can estimate age. This has been shown pretty consistently by multiple groups at this point. And this pan-mammalian clock was developed using over 11,000 CPG or methylation arrays, looking at 36,000 CPGs from 59 tissue types across 185 mammalian species. They showed that deviation from the predicted age correlates with risks and with interventions. There are specific cytosines that are conserved across species, and these implicate mechanisms that suggest an overlap between development and longevity, and mechanisms of cancer and obesity in there as well. Some of that is kind of interesting as you get into which genes are implicated by the CPGs and which pathways and mechanisms might be implicated by those. There are 3 clocks that are described. Clock 1 is kind of the original methylation clock, which regresses log transformed chronological age on DNA methylation. And clock 2 improves upon that by normalizing to maximal lifespan. So you end up with a 0 to 1 scale that, with 1 being the maximal lifespan, and now you can compare across species. Clock 3 further refines where instead of using maximal lifespan, normalizes to age at sexual maturity and gestation time. And this allows you to kind of correct for those cases where we may not really know what the true maximal lifespan of a species is. There are a couple of methods that are used to validate these models as they're created. First being leave 1 fraction out to demonstrate accuracy for estimating chronological age. This is similar to like a k-fold cross-validation in machine learning models. And with this, they showed a Pearson correlations in the range of 0.96 to 0.98, so quite high correlation, and median absolute error of less than 1 year on the predictions. Looking across species, they also did a series of leave 1 species out validation where you, for instance, leave Homo sapiens out, train on the methylation data from all of the other species, and then test that trained model back on the Homo sapien or on canine or whatever. And across that, the performance is a little bit reduced, but still reasonably high, and notably higher for the clocks 2 and 3. One of the challenges with the initial clock was that the correlation doesn't hold up when you use it to predict at species which have higher maximum lifespan, whereas that difference goes away with these new clocks. So looking here at a series of data that comes out looking at the validated universal clocks across species, we start to see these clock 2 and clock 3. Here, looking at clock 2 and here at clock 3, you can see that you end up with this range of 0 to 1 on the relative age, or in the log linear age, looking at the age of sexual maturity normalized. And then in both cases, you can transform that back into years and get the predicted years. They broke out marsupials here, which you can see are a little bit less consistent or able to be trained along with the other groups. And then looking at the leave one species out, you can see these clocks hold up relatively well. A few of the species are less less kind of strong correlation than some of the others. One of the things that I thought was interesting is they talk about the correlation of kind of 0.95, 0.98, like really high correlation numbers, and the less than a year MAE across everything. But then if you look at the Homo sapien alone, MAE of 4 years. So I think some of the species, human in particular, you see a wider range of error on that, which I think probably is indicative of the widely varying lifestyle choices that humans make compared to other species. So looking at the accuracy of the clocks and their relation to maximal lifespan, they took 69 of the species and then did correlation analysis. And for this, they normalized the lifespan to the maximal lifespan of bowhead whale, which is the largest at 211. And this is kind of a cool graphic that came out of this. You can see the dashed line is the maximal lifespan with species, bowhead whale being the longest lived mammal, and the cinereous shrew being the shortest lived mammal. And then you can see the maximal lifespan progressively getting up across the fold. And then here in red is the Pearson coefficient of clock 2, and in purple is the Pearson coefficient of clock 3 for the individual species. And you can see there's some variability across each of these groups. They work better or worse depending on which species you're looking at, but that variability doesn't seem to correlate at all with the maximal lifespan of that species. The background color here, the circumference of that background circle, is a Pearson coefficient of 0.9. And you can see everything's kind of, give or take around that center point. They then looked at the applications of this pan-mammalian clock in a few different perspectives. First, looking at a couple of human cohorts, the Framingham Heart Study and the Women's Health Initiative, a couple thousand participants each. And they showed that in both studies, using both clocks across both studies, the clock is able to predict hazard ratio for mortality risk, suggesting that as you deviate from your predicted age, or your predicted age predicts your mortality risk. They also looked at epigenetic reset. These are fibroblasts that have been reset with Yamanaka factors. And as expected, you see a reduction in the DNA methylation age with both of the clocks after the projected time. Interesting that the two clocks have slightly different time of onset. Maybe we're seeing earlier change, fibroblast and reprogramming being as variable as it is. I don't know how much I'd read into that, but they called that out. The next thing they looked at is interventions in mice and the ability of the clocks to pick up those changes. So this is kind of interesting looking at caloric restriction in mice, which is shown to reduce epigenetic age. And we see that indeed both clocks show a reduction in epigenetic age. Snell dwarf mice, which have lower epigenetic age compared to age-matched control mice, that shows up across the different tissues. And then growth hormone knockout showed also the reduction in epigenetic age, but conditional growth hormone knockout just in the liver does not show that reduction in epigenetic age, which is kind of interesting. So whatever benefit that is needs to be more systemic than the tissue. And then they looked at TET knockout, and two of the forms of TET showed epigenetic reduction of varying levels. TET2 does not show, and that matches prior reports of the effect of TET knockout on methylation age. Then here just looking more specifically at individual tissues. Okay, they then did a meta-analysis of the methylation change in the function of chronological age across species and tissues. Epigenome-wide association study identified 832 conserved age-related CPGs. One of the things that was noted is that there's a strong skew towards higher methylation with age compared to lower methylation with age. And then you start to see several highly changed CPGs or implicated genes emerging. They're going to talk about this LHFPL4 in a bit more detail throughout the rest. This is the CPG for that gene across 3 different species. So showing that again, the correlation of the beta values correlates with the age of the animal. Did breakout comparing marsupials and monotremes to eutherians. There wasn't a lot of description in the text about this, and I didn't catch anything that looked terribly different. But I think, going back to the other correlations where these clocks aren't quite as accurate for those, I think there's maybe some attempt to figure out what's going on there. And then looking at tissue specificity of the CPGs across different areas, of the top 1,000 or so CPGs that are different between young and old or driving these clocks, there's not a ton of overlap with these, but there are 51 implicated genes, I suppose, that are different, that are shared amongst all of those. Almost all of them are positively correlated, so increase of methylation with age. They then took those top 1,000 sites that change with age and looked at them across different tissue types, looking at I think these are the mean methylation of those sets in this tissue and the extent to which they go up or down. Interesting thing that emerged here is that the patterns are different between proliferating cell types like blood, skin, and liver compared to non-proliferating cell types like muscle, brain, and cortex. This is something that's kind of been running in the back of my mind for a while as well, that we have 2 very distinct types of biology happening with association to age with rapidly turned over cells versus very long-lived cells. And I think digging into these methylation differences with age could be very informative. So I'd be curious to dig into that or hear other people that are looking at that. One of the things here that they talked about with this data is that the positively age-related CPG sites were significantly enriched in states associated with polycomb repressive complex 2 binding sites, which is a transcriptional repressor complex. I'm not familiar with that biology myself enough to really explain what that is, the implication of that, but I think that may lend some clues as to the aging mechanisms there. I'm curious if other people have comments or have looked at that. Looking at that single CPG again in depth for mouse tissues, I won't go too heavily into this, but they showed mouse in development, looking at various tissues, and see mostly the correlation holds, not so much in blood, but the others seem to be holding up relatively high. And then they also looked after development. Interestingly here, where blood is a little scattered there, much stronger correlation in blood after development. So we see some differences there. And then you can see the correlations here. You get into tissue specificity again, and less than 0.5 correlation in most of the brain or the macrophages, which is kind of interesting, whereas you see the strong correlation in a lot of the peripheral tissues. They then looked at epigenome-wide association of age in different age groups. And so bucketing ages, is interesting just to see how this holds up over the development and timeline of life. Looking at 3 buckets, young defined as 1.5x of age at sexual maturity, and old as more than 3.5x age of sexual maturity, and then everything in between as middle. Looking then at the correlations here of young versus middle, old versus middle, and young versus old. The key players are maintained across all of these comparisons, a little bit of shuffling around, but the correlation seems to be relatively conserved across those age groups. And this pattern talked about where you see a trend toward more methylation as you go from young to middle to old, and that changing throughout lifestyle. Looking then at biological pathways and functional analysis, they use 3 different methods here using the genomic region-based GREAT tools, transcript-wide association studies, and genome-wide association studies to compare the methods here. Few things that were interesting. There are obviously mortality and aging having this pretty high correlation with increased methylation of these sites. There are cancers up there and other things. The one that caught my eye as being weird is Alzheimer's disease, where you see an increased methylation in blood and liver, but decreased methylation in muscle, brain, and cortex. So that association showing that with Alzheimer's disease, you may have 2 very distinct mechanisms going in peripheral or proliferating tissues versus brain and non-proliferating tissues. And maybe there's kind of what the shared mechanism or that might be, or the relationship between those, I think is quite interesting. Almost everything going up, but the few that do go down kind of stand out. Alzheimer's being one of them in the brain. Circadian rhythm also seems to be driven by age-related reductions in methylation, and cancer also in the brain and muscle, these same tissues, looking at reduced methylation. So that is that. Then the next thing we looked at is single-cell ATAC-seq to look at open chromatin of a driver of maybe expression changes. And you see that of the top over-methylated sites, they are associated with reduced open chromatin with age for many of these genes. Looking here again at this LPFPL4, you see that ATAC peak call getting reduced with, or more peak call going up with age, suggesting that there's the reduction in levels there, I believe. Looking at specific cell types, the increased call seems to be more specific to stem cells and progenitor cell types compared to others. And then they looked specifically at the subtypes here, and the only one that has a significant correlation is the hematopoietic stem cells. Progenitor cells and differentiated cells do not seem to have the chromatin signal with that. They then validated that looking at hematopoietic stem cells in mice. And when they did that, they did see the old-young correlation, but the specific CPGs and implicated genes were different in mice than they were in the human HSCs. So they didn't go into that a lot deeper, but there seemed to be mouse-human differences there. So in conclusion, these clocks do seem to be highly conserved and very accurate with the proper transformations and controls. We're now getting to clocks that can work across species. The clocks, while they are conserved across multi-tissue, specific CPGs seem to differ by tissue and cell type. Proliferating versus non-proliferating in particular, I think is super interesting. And I'm curious to see more work go into that. And one of the key takeaways here is that the mechanisms of development and aging seem to be very intertwined. Kind of the same things that are activated and changed during development kind of subside and then get reactivated during later aging, which is kind of interesting. So I think there's there's a ton of data underneath of this to dig into. Also, in line with the way this group has worked before, all of the data is publicly available. All of the code is available on GitHub. We've been able to replicate the prior clocks in-house pretty easily using those methods. So I think this is helpful to have these tools, not just for age prediction, but also for application to other forms of disease.